Extreme drought is one of many impacts of climate change. Globally, we've seen droughts increase in duration and intensity, with many negative impacts on natural ecosystems, crops, and our economy. This begs the question, how do plants deal with drought?
Our guest today, Dr. Jarmila Pitterman, studies plant ecophysiology, how plants are structured and what that structure means for how plants respond to their environment. She seeks to understand how different plants use different strategies for surviving drought by zooming in to look at their internal workings, their plumbing. Her exploration of the various systems plants have developed for moving and using water is showing us that some plants use some pretty unique methods for dealing with drought. Let's explore nature's creativity with Dr. Jarmila Pitterman. This is RadioBio.
Don't know much biology.
Hello and welcome to RadioBio. I'm Kinsey Brock.
And I'm Jeff Lauder. We are joined here today by Dr. Jarmila Pitterman, a researcher at the University of California, Santa Cruz. Can you please tell us a little bit about yourself? Who are you and what do you study?
All right. Well, I am an associate professor at UC Santa Cruz in the Department of Ecology and Evolutionary Biology, and I am a plant ecophysiologist, and my particular focus is plant water transport. Plant water transport, along with photosynthesis, is one of the most central aspects of plant function, and so I try to understand how plants transport water while maintaining some resistance to drought stress or freezing stress. Both types of stresses can impact water transport, and so it's been very interesting to learn about the evolutionary, ecological, as well as the applied aspects of plant water transport.
Okay, so ecophysiology, that sounds really cool. You kind of described what that is a little bit. So how did you get into studying ecophysiology of plants?
I got into ecophysiology because of, mostly in some ways, serendipity, but it was probably meant to happen. I met the right person at the University of Toronto, and he took me under his wing and was a great mentor to me.
Awesome.
And that then propelled me forward. So that's how I got into it. And I think maybe to go back to the original question, which is like, why in ecophysiology? And I think it's because I was always interested in wanting to understand how organisms work. You know, my father is a mechanic, very process-oriented mind frame, like how do things work? And I think I inherited some of that. Or anyway, it certainly seemed apparent to me that that's the route that I was going to take, is trying to understand how organisms work. Mm-hmm. And plant ecophysiology was just the right venue for me. And so it was a combination of mentors, just inherent interest, and some luck. Yeah.
So speaking of kind of the mechanics perspective, and you mentioned in your description of what you study, water transport. Mm-hmm. So what does that mean? How do plants move water?
So, well, plant water transport is one of the most essential aspects of being a terrestrial plant. Without an efficient means of transporting water, plants would desiccate and dry out in the atmosphere. So there's great evolutionary pressure on optimizing that system. So plant water transport is actually pretty simple. It's we call it parsimonious. It's a cheap process that transports water from the soil through the root into the xylem and out through the leaves into the atmosphere along what we call the soil-plant atmosphere continuum. And the process is passive in the sense that there is no metabolic energy expended to move water throughout the plant.
Dr. Pitterman mentions water transport is passive. What does that mean?
Well, she mentions all plants have pores on their leaves called stomata. Plants open these stomata to take in carbon, but they lose water. When they lose water, that creates negative pressure that pulls water up through the roots and into the plant's plumbing, which we call xylem.
It's the atmosphere that's pulling on one end, and it's the soil that's pulling on another, because water adheres to soil particles, right? And so that water moves from the soil into the root, throughout the xylem, and then out through the leaf. It's more of a combination of wicking, capillary suction, pulling up a continuous water column throughout the plant. Basically, water moves from a place of high hydration or from wet soil to the dry atmosphere through the plant. The plant is really a wick for this. And it's a necessary evil of doing photosynthesis. 98% of the water that's moved through transpiring trees is moved through the stomata to enable photosynthesis. Less than 2% of that water is used in biochemistry and metabolism.
All plants breathe carbon. That's what photosynthesis is. They take in carbon through their stomata, but like we said earlier, there's a trade-off between taking carbon in and losing water. This trade-off becomes more intense under harsh conditions like drought.
So California just experienced one of the most intense droughts in a really long time. How does drought affect plants in general?
We're actually just starting to learn about that. Oddly enough, it seems like a very simple question. How does drought affect plants? And especially, how does it lead to mortality? And we're just starting to get a handle on some of the predictors. Plants can be, like I was saying earlier, they can be differently adapted to it. But what we're learning is that short periods of drought are not as deleterious, not as detrimental as prolonged periods of drought. So plants can recover from short episodes of drought. But if you have these long 5-10-year episodes of water deficit, that really does start to impact plant growth. Parts of the plant will die off. We're talking about trees here. So parts of the canopy, usually the top parts of the canopy, will be the first to dry out and die. Growth rings will be a lot smaller in trees. They're not going to put on as much radial growth. Photosynthesis rates are going to be a lot lower. They could be holding on to their water with stomatal closure. I wish there was a really simple, glib answer that could tell you, okay, well, if you don't water a plant, it dries up, loses hydration, leaves fall off, and it dies, right? But in plants like conifers, the process is more prolonged, and then there are episodes of potential recovery, so
you're right, because when you talked about short periods versus long periods, naturally, when you think of California has a dry season. And the trees don't die, the conifers don't die, the ferns don't die.
Right.
But if it's over several years where they're not getting what they need, then we see this really detrimental effect.
Yeah, absolutely. And the plants just seem, they're alive, but they seem tapped out. That's kind of my impression of the plants around Santa Cruz, after this prolonged episode of drought, is that they just weren't putting on as much growth. They were showing dieback. And I think some of that could point to carbohydrate depletion or sugar depletion when they're just not photosynthesizing enough to replenish their carbohydrates and their sugar reserves.
So in order to grow, plants need the carbon to make carbohydrates.
Yeah, during drought, plants take in less carbon, meaning they make less of these carbohydrates, including sugar. When this happens, the plants are basically starving and don't have enough raw material to make things like new plant tissue.
They eventually can't grow as much. They cannot invest in protective compounds, right? So, they become more vulnerable to pathogens and to insects. The plant has to, and I hate to use this term, but I'm going to use it, as decide on how it's going to use its limited carbon resources. Is it going to make seeds? Is it going to grow? Or is it going to protect itself, right? Mm-hmm. So how do those three aspects of plant function become priorities, during or after drought? Mm-hmm. But what we do see is that drought-stressed plants become more vulnerable to pathogens, and we think it's because their carbohydrate reserves are depleted.
So this trade-off is important in all plants, but how do different kinds of plants respond to drought stress?
Do different species of plants have different physiological responses to drought?
Plants have had to deal with drought stress ever since they made it out onto land, right? So, it's a pretty important factor in being successful land plants, being able to handle drought. And being able to transport water efficiently. The first land plants were probably just simple little like liverwort-like thalli, and they were tolerant of complete desiccation, and then they could rehydrate following rain or immersion into water. But if you think about how plants evolved, they would have started to compete with one another, and they would have started competing for resources such as sunlight, for example, and propagule dispersal, and so that would have prompted them or selected for them to grow tall, right? And once you have a plant that's more than a centimeter or two in height, it has to evolve an efficient way of transporting water by some specific tissue, because just movement of water from cell to cell is inefficient. So plants have evolved this tubing, the xylem that we've been talking about.
What about different perennial plants versus annual plants?
Plants can adjust in a number of ways. So herbaceous plants can adjust. So they can delay seed set, they can delay germination until there's sufficient water available. And they're annuals, and they can complete their life cycles faster, so they're more functionally flexible than long life perennials like trees, right? So, the response of a tree might be more subtle, and trees can also store significant quantities of water in their wood, so they're a little bit more buffered against drought, and so they can resist for a while. But like I was saying earlier, if the drought is prolonged, then that buffer runs out, right? Then their safety margin gets narrower and narrower, and their xylem begins to lose transport capacity, and the phloem becomes compromised. So yeah, when drought strikes a tree, a lot happens that can all conspire to really affect that tree's physiology. So
you talked about drought affecting transport capacity. So what are some of the things that physically, like what would block water movement in a plant?
The process of water transport means that in plants, water is transported under tension, and so that water column is under negative pressure. And that means that if there's some kind of disturbance, a wind breaks a twig, or an herbivore chomps into a leaf or part of a stem, that allows air to enter the xylem, okay? And so whenever air enters the vascular tissue, that vascular tissue can no longer transport water. In situations of extreme drought stress, which mean that the loss of water from the canopy is greater than the ability of that plant to replenish that water to the canopy, you get the water in the xylem develops tension. And different species can tolerate different tensions. They're evolved for that. So, for example, a plant with sufficient access to water is not going to be tolerant of excessive drought. It's not going to be able to handle this tension in its vascular column in the same way like a juniper, for example, or drought, more drought-tolerant species.
It's just used to sucking up tons of water.
Right. And so in the riparian plant, for example, what's going to happen is that air is going to start entering into the xylem conduits, because that xylem tissue is not going to be able to handle that negative tension, that pressure, that negative pressure in the cells. And so air is just going to start entering those xylem conduits through injured areas or lesions in the cell walls that normally wouldn't be a problem but are now a problem because the plant is under this drought stress. When you have a conduit that's blocked with air, it can't move water. Yeah. And so it's the same situation that happens, I guess, in humans, right, with embolisms in our vascular system. It happens in plants too. And under situations of great drought stress, those embolisms really add up to the point where they block water transport. And some plants can recover from embolisms, others can't, right? So conifers do not have a means of recovering from embolism other than by growing new growth rings. Mm-hmm. That's it. Whereas some angiosperms do have that means, and it's a topic of great interest, to growers. There's a lot of work being done on grapevine, and the ability of grapevines to refill air-filled xylem conduits, right? Mm-hmm. And so there actually appears to be active metabolic input in that recovery process in vitis in grapevine. So that's a topic of a lot of interest right now. A lot of debate is like, how do plants actually refill. But some plants we know just don't, or the evidence is just scant for their refilling, or it's not completely understood.
How do you actually study this stuff? How do you study like how water moves through a plant? Like how do you actually study xylem physiology?
Right. Well, there's a number of ways. So at the leaf level, you can measure leaf water loss, right? And that can be done with what we call a gas exchange instrument. And so it's basically an instrument that measures how much CO2 the plant takes up and how much water it gives off, right? So we can use that to infer how much water the leaf is losing. As far as studying water transport, in some plants it's easier than others, but basically if you want to have a sense of how efficient, let's say the xylem of Pinus ponderosa is, then what I would do is go in the field, I would clip a branch, bring it back with me to the lab, hook it up to some plastic tubing, and then measure rates of water flow through that stem. That's a really simple way of doing it, but that's what we do, is we treat the xylem like pipes, right? And, the assumption is that the smaller the xylem conduits, the slower the transport will be, and the larger the xylem conduits, the faster the flow rates will be. So, for example, if you were to measure water flow through something like grapevine, which has huge vessels that can be 0.5 millimeter in diameter, they have incredibly high rates of water transport. And then compare that with something like drought-adapted cedar, which has really tiny cells, then that's going to have very low rates of water transport. So that's, so literally going out into the field, clipping stems, and then treating them like plumbing, right? So that's another way of doing it. And then what I like to do in my lab as well is I like to look at xylem anatomy, and from the size of the conduits, infer how much water they're capable of moving. We can model that. It's not perfect, but it's pretty robust, and it can tell us a lot. And it's powerful because it can be used on non-living plants. It can be used on growth rings. It can be used on fossil plants.
Cool.
Yeah, so it can be very revealing. You can learn a lot about a plant's physiology just by looking at its wood. There's a lot that you can infer from that. So we take these kind of three approaches of field measures, lab measures of transport efficiency, and then anatomy. So those are the three basic approaches. And of course, each one of those is nuanced, and you can do other things, but that's basically it. Yeah.
Dr. Pitterman's research interest in drought physiology has taken her to two different kinds of organisms, conifers and ferns. It turns out the difference in water transport between a woody plant and a non-woody plant can tell us a lot about how plants survive drought. We've been talking about ecophysiology, and you study the ecophysiology of conifers. What are conifers?
Conifers are all trees or shrubs, actually. They're woody plants. They produce seeds, but they do not flower. Okay, these are non-flowering trees. That's the big distinction between conifers and angiosperms. The wood, which is really the xylem or the water transporting tissue in conifers, is pretty simple. It's made up of one single cell type for the most part, called a tracheid, and those are empty, dead cells, but they're functional when they're dead, so I call them zombie cells. And then they have some fraction of living tissue as well, known as parenchyma. And in that living tissue, we see a lot of storage of starch and proteins and other elements that a plant will need to survive. So anyway, getting back to your original question, so conifers, they have a very different morphology than angiosperms. Strong leader shoot. They don't branch in the same way. Oftentimes they don't branch in the same way as angiosperms do. But the big difference is that their wood is made up of tracheids and that they do not produce flowers.
And so you also study ecophysiology in ferns.
That's right.
So what are ferns and how do they differ from conifers?
Well, interesting question. So ferns are vascular plants, so they do have xylem and phloem. but they do not produce seeds, right? So in that kind of tree of life, they are more ancestral to conifers. They do not produce seeds. Instead, they produce spores on the underside of their leaves, and then those spores go on to produce reproductive propagules. Modern ferns, including tree ferns, do not have wood, and that is a very important constraint on the success and the biomass of ferns, in that they do not produce wood. So structurally, it couldn't be more different than a woody plant, and that's their impediment because they do not have a cambial layer like woody plants. They do not make lateral branches, and one of the missing pieces, I think, of ferns, is one of their problems, if you could call it that, is they can't make secondary xylem, and so they've had to figure out ways of dealing with that and exploring niches in different ways, despite these morphological constraints.
How do we think of the structures of a tree versus a fern differently, in terms of ecophysiology and drought response?
Ferns have tracheids, and tracheids are, like in conifers, they're single cells, basically, right? Not like vessels where you have stacks of cells making up a long tube. No, they have tracheids. Those, tracheids are bigger and longer typically than those in conifers. They're not as mechanically reinforced because it's not wood. It's not holding up the frond, right? Mm-hmm. That's achieved using a different type of tissue, these different fibers. So, ferns have what we call as primary xylem, and that's it. Secondary xylem is wood. Mm-hmm. Primary xylem is what you have in herbaceous plants, like basil, for example, mm-hmm. that's primary xylem. That's all ferns have, right? They cannot make wood. And so that's a good thing, maybe, because they have some flexibility in how they develop their leaf morphology, for example. They're not canalized in some ways. Maybe they are in others, I've always thought that not being able to branch was a big impediment, because if you think of something like an oak tree where you have extensive lateral branching, those branches can grow toward light. Right? In ferns, it's just not possible. Right? There's only so much extension that a frond can do unless it's adapted, specifically. So I think not being able to branch is a pretty significant impediment. But on the other hand, they are super diverse, 13,000 species. So what is it that they're doing right? We're learning about the physiology of these plants. Learning really about their ability to recover from drought too. That's teaching us a lot. Mm-hmm. There are many fern species that live in the deserts, and they're what we call desiccation tolerant. So they can survive losing over 95% of their water. They're adapted to that. They go into this quasi-dormant state, and then once the rains hit again, they rehydrate and resurrect and become metabolically active once more. And
it's like an amazing recovery, isn't it? Like they can lose up to 95% of their water, but then they come back. And they're totally cool.
Yeah, some of the cells, some species it'll vary, but the cells can enter this kind of glassy state where the cells just become so full of sugars and various protective amino acids that immobilize membranes and proteins to prevent them from denaturing.
So when these ferns dry out, their cells almost enter this type of cellular hibernation, which Dr. Pitterman here refers to as a glassy state. The cells turn more solid and wait until rain gives them the signal to start growing normally again.
It's an incredibly expensive process, right? This is why it's only small plants that can do this. It makes no sense to have a desiccation tolerant sequoia, right? Because that would be metabolically just so expensive.
Sequoia cannot do that.
Right. And in fact, sequoias have wood, right, that transports water efficiently. So there's no need to have that. It would be hyper redundant to be able to lose 95% of your water and also have this incredible hydraulic system. When you're looking at drought resistance in conifers and angiosperms, you're looking at nuances, right? So how does that tree retain water? So there's kind of a range of physiological responses, right? But what we're seeing in ferns is like completely new things, right? New ways of structuring their xylem, new ways of arranging their phenology. Ferns are important entities in redwood forests and understories. And so when you walk through a redwood forest, most of the time you're looking at the understory and you're looking at the ferns. And when those plants are responding to drought by either, dying or losing fronds or curling up, that there's something going on.
Yeah.
And, that's what we've been seeing in coastal California. And that's what prompted this research into studying fern drought responses in the redwood forest. It's also easier to work on ferns than to climb redwoods, right? And so, but the idea is that by looking at the understory, that might give you a clue as to what's happening at the top of the redwoods. So that's another interesting aspect of this. Because I think science and plant science in a way is kind of moving toward, it's scaling up. It's like we're trying to understand systems and how systems are going to respond to water deficit. Yeah. So you can make it as granular as you want, but in the end, you're going to have to look at the big picture of the biome. And so I'm hoping that by looking at the fern understory, that will add another important component of information to understanding the redwood forest as a whole. And it has practical applications too, because my funding agency, which is the Save the Redwood League, uses that information then to make critical decisions about which parts of the forest to sell off or conserve. Or is it close to a stream or a water source? So understanding how those plants respond shapes decisions.
Yeah,
I think.
Decisions about environmental management are becoming increasingly difficult to make. We have to plan not only for immediate concerns, but long-term climate change impacts.
Climate change projections predict extreme shifts in temperature and rainfall. Studying how plants respond to drought can help us understand how natural systems respond to drought, but also can have other implications.
It's, I think, opening a lot of doors of inquiry that could potentially be useful in crop applications and understanding plant responses to drought overall, understanding ecosystems as a whole, right? Yeah. There's a lot of interest in understanding the physiology, the physics, and the molecular biology of that process, because you could see that it has practical applications to crop development, for example.
Oh, yeah.
If there are genes that are especially critical for drought tolerance or recovery from drought, then it would be useful to know that and to consider genetically engineering those genes into crops, for example. But I like to think big, and I like to think about processes and telling stories, right? I like to think about how I can tell an interesting story about the evolution of ferns, about the evolution of conifers, right? I'm interested in species biogeography. I'm interested in understanding how physiology constrains, biogeographic distributions and how that's going to be impacted by climate change.
Dr. Pitterman's research has implications for how natural and crop systems respond to drought. We thought we had a good understanding of how plants respond to drought, that all plants respond similarly, but it turns out that's not true. Dr. Pitterman's research into ferns is showing us how much we truly don't know, and how studying unique organisms can sometimes be the best way not to only find new answers, but also generate new questions.
What's interesting is, how these ferns recover from that stress, right? There's a lot of DNA repair, a lot of protein repair that happens. These are all expensive processes. But I think that these are mechanisms that have not been investigated as thoroughly in ferns as they should be, because they might give us ideas, right? I think nature has all sorts of secrets, and there's a lot to learn. And I think that by looking at some of these taxa that haven't always attracted attention because they're not as charismatic or they're not crop plants, they can still reveal a lot about plant function that I think would otherwise be overlooked, and that perhaps we wouldn't have the creativity to see for ourselves, right?
Yeah.
So I feel like let's explore nature's creativity and see what's out there.
I want to talk about nature, yeah. We got nature everywhere you go. Nature on the street, nature in the disco.
There's a whole world of questions to explore in plant ecophysiology. Dr. Pitterman leverages different plant systems to understand the basic form and function of structures related to water transport. It may seem like a simple thing, but we're just starting to uncover how plants deal with drought, which is important to understand in a changing climate.
This has been RadioBio with Dr. Jarmila Pitterman. Thanks for listening.
RadioBio is produced by graduate students at the University of California, Merced. Support for RadioBio comes from the Quantitative and Systems Biology Graduate Group, the School of Natural Sciences, and the Graduate Division at UC Merced. You can help support RadioBio's mission of increasing scientific literacy in California's Central Valley and beyond by donating at giving.ucmerced.edu/radiobio. Find out more about our mission, events, and podcasts at www.radiobio.net. This episode of RadioBio was produced by Suninta Pertiwi. The interviewers were Jeff Lauder and Kinsey Brock, and the episode was edited by Jeff Lauder. The artwork was created by Jeff Lauder.